IPs. And we have to march for the Skill India and Startup India. So we are here. I do welcome for this August gathering, which is being chaired by today's guest speaker, Dr. Sarika Gupta ma'am, who is scientist from National Institute of Immunology. She will be delivering and she will be enlightening us with respect to the topic title as Protein Aggregation from Disease to Long-Acting Therapeutics. So, I do welcome you, ma'am. I do welcome Honorable Principal of our college, Dr. Bharat Shinde sir. And because of his health, he could not join today's session. He was willing to, but could not. I would welcome officiating principal Dr. Shamrao Khadge sir, vice principal of our college, Dr. Lala Saib Kashis sir, Dr. Tushar Bose, who coordinates for IIC and who is director for Indira Gandhi National Open University courses also. So, ma'am, I would like to tell to our students and participants who are enrolled at this particular online platform that this particular Vidya Pratishthan's Arts, Science and Commerce College, it was established in 1994 with just meager strength of 150. And now after 26 years, we are having 6,000 plus students who are being getting educated up to their PhDs. So, right from KG to PG, the campus, which holds around 30,000 students under the banner of Vidya Pratishthan, which is referred as the Oxford of East, presently in Maharashtra especially, and who is pioneer in higher education in Maharashtra, especially with respect to biotechnology as well as information technology, if it is being concerned. We do have PhD research center in biotechnology as well as physics subject. We do have a research institute with the Pratishthan School of Biotechnology as well as agriculture biotechnology. And for Indira Gandhi National Open University courses, we do have around 200 plus students. So with such a strength, we started in 1994 and we have been accredited by NAC with A plus as well as B double plus for third cycle. And we do have been honored by as well as ranked by MOE, Ministry of Education, with respect to Atal ranking as in the first 25 colleges, institutions nationwide. And this year we are the band performer also amongst the non-technical category. So it's a great privilege for us so as to welcome you all in this particular gathering. Yes, we are all really very eager to listen from Dr. Sarika ma'am, as we all, and I do welcome one and all again on this our campuses virtually at Vidya Pratishthan's Art, Science and Commerce College. So hereby, I would like to tell you that about DBT Star College scheme. Dr. Bose sir, Dr. Chafalkar ma'am, they had filed for DBT Star College scheme in 2009, and up till now, it is the only college campus across the nation. Uh, just from the inception of the scheme, we are with DBT India. So presently, from Star College, we march towards Star College status also, and now presently we are mentoring college. So because of so much of activities and enthusiasm from the students' part, so DPT had taken this concern and have taken us among the first 50 colleges across the nation with respect to DPT funding. So because of this, we could also fetch the grants for infrastructure development under DST FEAST also. And due to which we could begin with the few professional courses like we work in food technology, and under one umbrella, food technology, microbiology, biotechnology, agriculture biotechnology, such degree programs they have been undertaken, and it is one of the school with respect to the research concerning this subject which has been going on, and because of which we could fetch such a ranking, and it has been possible because of our students. So, ma'am, as well as the participant, with a very proud feeling, I do feel that I'm a part, and we all are part of Vidya Pratishthan. And where sky is the limit, we are encouraged by the visionaries like Honorable Sharad Chandraji Pawar Sahab, who is Padma Vibhushan, and under his guidance, Management Council, Governing Council, Executive Committee, as well as Mrs. Sunitra Tai Pawar, we all could gather such particular events, the academic events, and our endeavors, they do fructify. So aptly, if we were to say that the Candid Lush Green campus around on the 200 acres, so Lush Green campus, it has been audited with energy audit, environment audit, as well as green audit, as well as gender audit also. And we do have as an institutional distinctiveness the Nakshatra Garden, for which more than 4 patents have been sanctioned for this particular facilities, under which the work it has been consideration in the phase one, and almost more than 200 papers they have been published under this banner of medicinal plants. So it's really a great privilege and the legacy which has been continued from the Department of Microbiology as well as Department of Biotechnology, especially and Food Technology. We could march our life sciences students for this particular inventions and discoveries. And so today's topic, it is again related to such platforms. I would like to say that how our lab on the bench, what we are going to study, what we do study the proteins, how they are applied. So it is a translational research that for long acting therapeutics, how we can march ahead. So it will be really great honor, ma'am, so that we'll be enlightened with your kind words. So on this Azadi Ka Amrut Mahotsav, I do once again welcome you all, and I do really appreciate the work by Dr. Borse sir for arranging and taking this painstaking efforts for organizing this particular. So thank you very much.
Thank you very much, ma'am, for those kind and warm welcome words. Now I request Dr. Tushar Borse, sir, Coordinator, Deputy Star College Program, to introduce our guest of honor. Please, sir.
Yeah. Good morning to one and all, respected dais, today's guest speaker, Dr. Sarika Gupta, and my dear friends. Today we have gathered here for an online webinar on the occasion of Azadi Ka Amrut Mahotsav. So we have our guest speaker, Dr. Sarika Gupta from National Institute of Immunology. So it's my great honor to introduce her. So Dr. Sarika Gupta has completed her MSc and doctoral degree from BHU. She has done her postdoctoral studies at IISc Bangalore, and presently she is working as a staff scientist at the National Institute of Immunology, New Delhi. Her area of interest has been protein misfolding, neurodegenerative diseases, inhibitor designing, then chronic diseases, which has fetched her different awards and recognitions such as Srimati Kusum Sharma Awards in 2014. She is also a recipient of SERB Women Excellence Award in 2013. She has also been awarded as Young Scientist in 2011 by National Academy of Sciences. She is also working as a visiting scientist to the University of Miami, USA. As a life scientist, she has worked a lot in the field of protein, and to recognize her work, she has published a number of papers, and she also has some patents to her name. Okay, so she has licensed some industries for their commercialization. So with this short introduction, I welcome Dr. Sarika Gupta to deliver her lecture on protein folding and its uses. So it's one of the topic of my interest personally, because I do love proteins, and maybe the students, my students who are from the field of biotechnology, microbiology, zoology, to whom I'm continuously delivering some points about the proteins. Maybe this could be an additional things what they could learn through Dr. Sarika Gupta. So with this short introduction, I welcome you once again, madam, so to deliver your lecture. Before she starts, I'll request Dr. Meenakshi Munshi, who is a consultant at NII, to introduce about NII, and then we'll continue with the lecture of Dr. Sarika Gupta. One more announcement which I'd like to make for the students, that at the end of this session, we'll be having a small quiz which will be organized. So be attentive, listen to the points what madam is going to cover up, and a small quiz will be conducted at the end of the session. Right. Okay. Yeah, Meenakshi madam.
Thank you, Dr. Borse, for this wonderful introduction of your college and I would share my screen in a minute. One minute. So, okay. This is, I need to share my screen. So how does it work?
Yeah, I think the screen is visible.
Oh.
But the presentation which you want to show, you can.
But I can't see my, presentation. I have to, 1 minute, maybe I will stop the presenting and then first I have to. So this is. One minute, I am trying to. But I see your screen. It is not my screen. That's the problem.
Ma'am, please open your keep your presentation ready on the desktop first.
Okay. First. All right. I'll do that. Is it, can you see that? No.
Ma'am, you have to first open your PPT, then you have to start sharing.
1 minute. I have opened the PPT. PPT is open.
Yeah, and now you come on the this platform, Google platform where we are having the meeting, and then you can start sharing.
Okay, 1 minute. Where do I see this meeting? Because if I put it in minus and then it goes in the minus. Okay. Can I share it? I don't know. I'm trying to. Something is wrong somewhere. No, again, I can see this only. No. Jyoti, can you help me in sharing from your screen?
Yeah. What I can do is that I can share it from my screen and then you can start.
Yes. I think that's better. Sorry for this goof up.
Is it visible?
Yeah, You can put it in the slide mode.
It's in the slide mode, ma'am.
But anyway, good morning once again to each and every person who has joined this meeting. And before Dr. Sarika Gupta starts her presentation, I would like to tell you brief about our National Institute of Immunology, where she has done this excellent research, which you are going to hear in a short while from now. National Institute of Immunology was, in fact, it was started in way back in 1982 as a part of the Department of Science and Technology, and eventually it became an independent institution of Department of Biotechnology way back in 1986. Can you go to the next slide, Jyoti?
Ma'am, is the next slide visible to you?
No.
Are the slides moving?
Yeah, now it's moving. Well, the mission of setting up of this institution had been to undertake a research, aid, promote, guide, and coordinate research of high caliber in basic and applied immunology, and to provide and promote effective linkages on a continuing basis between the various scientific and research organizations and laboratories and other organizations which are working in the country in the field of immunology. And also one of the important mission is to organize...
Madam, you are not audible.
Am I not audible?
No, the voice is too low.
I don't know. I'm speaking at the pitch of my voice. 1 minute. My volume, is it okay now?
Yeah, it's okay now.
Okay. So, one of our aim is also to organize postgraduate courses, workshops, seminars, symposiums, and training programs in specialized, especially in the immunology area. And that's how we are here today. Can we move to the next slide, Jyoti? Jyoti, next slide.
Yeah, ma'am. It's the next slide.
Goals.
Is it?
I don't see. We come to the goals next. Is what are the goals of our institution? Yeah. See, when you talk about the goals, the broad goals of National Institute of Immunology. What's happening? Someone else is presenting. What is this? Guriya Kumari is presenting. I lost my slide. Jyoti?
Ma'am, no, ma'am. Yeah, there is something.
There is some Guriya Kumari is presenting.
Miss Guriya Kumari, can you please stop your presentation?
Stop your screen. Can you remove
Miss Guriya Kumari, can you please stop your presenting your screen?
Sir, if you have the controls, can you please?
No, I don't think so that I can control here. I'll just check.
Otherwise, can remove her, no?
Can we do that?
Yeah. Yes, I removed her.
Oh. Jyoti, are you now able to share?
Ma'am, I'm doing it.
Yeah. Sorry for this technical glitch. When we talk about the goals of our institution, it's to provide a national and international leadership in the immunological sciences, provide quality healthcare solutions to the country's major medical problems, and expansion of immunology science by incorporating tools and methods of chemistry, physics, mathematics, and other allied sciences, and play a leading role in creation of immunoengineering. Next, Jyoti.
I think there's some problem, ma'am. Can you see the next slide?
Yeah, I can see that. See, these are some of the activities which have the research done in the lab has moved outside the lab and really reached a large number of people. We at NII has been able to develop anti-leprosy vaccine, anti-cancer immunotherapeutic dendritic cell-based vaccines, product development, and cell and intellectual property rights cell, which provide vital support in efficient transfer of research to the industry. And these are some of the basic vision and the goals of the institute. And under this, today's top lecture by Dr. Sarika Gupta will enlighten you what she has been doing in the lab at NII. Over to Dr. Gupta. Thank you, Jyoti, for sharing this.
Thank you, ma'am.
And I would like to also share that I had been a part of the Star College scheme in the department prior to joining NII. I worked as an advisor with the Department of Biotechnology, where I was handling this Star College scheme. And you all know, you have been a part of the Star College, how successful the scheme has been over the years, and it has created a brand in itself. And now we want the college like Baramati, this college should mentor, as you have been mentoring the other colleges, and mentor more and more colleges, and come to DBT and expand this family of Star colleges. Thank you very much. Over to Dr. Gupta.
Hello, good morning, everyone, and thank you, Dr. Tushar and Kalpana and VP ma'am for a kind introduction. And I feel that I will keep all the audience. Am I audible and the screen is visible?
Yes, ma'am.
Okay.
So I would request, before you start, you also once again tell the students that please listen the lecture very carefully, because at the end of the lecture we'll be having a short, very small quiz questions for the students. So yeah.
Okay. So as I will switch off my video so that there is no problem while presentation. So student, protein aggregation is the biggest problem, and it has been linked with the disease since long back. And we are for the first time, we have utilized this as a tool to develop a long-acting therapeutics. And I hope that I will be able to make you understand how it can be done. Jyoti, my slide is moving or not?
No,
it's not moving, Dr. Sarika.
Again, I'll share. Okay. Okay, so before going into my topic of lecture today, I just wanted to introduce you about the work that we are doing currently in the National Institute of Immunology. My lab name is Molecular Sciences Lab, and this is the my students and the whole team. So in my lab, we are concentrating on two different organs, like brain and the bone. So in the brain, we are mainly wanted to study the neurodegenerative diseases, where we are concentrating more on the Alzheimer disease, Parkinson disease, and prion disease. Basically, how the initial phases of pathogenesis, so that we can identify new targets and develop a therapeutic for them. In the bone and joint disease also, we are working on how the process of bone remodeling is regulated, and can we develop target some of the pathways in the bone remodeling process to develop drug, and also how these two organs they communicate with each other. So we are working on a very complex problem, and definitely doing a very interesting finding. So today we are going to talk about how protein they are important in the cell, and if they're folding, they are not properly folded, and they can cause different kind of diseases. So we all know that in our body we have because protein needed to they are performing so many vast kind of function, and they are involved in very specific interaction with like you can say different kind of process in the cell or body. So cell has a very tight quality control for this protein. You can understand that once the protein is synthesized into the AR, they are correctly folded. If they are correctly folded, then only it will be transferred to the other part of the cell, or you can say organ, to perform their function. If these proteins are not properly folded, or you can say they are in the misfolded state, they are identified by the tools in the cells, and those proteins specifically, they get degraded by different degradatory mechanism of our body. So to understand better what is this folding and unfolding that we are talking about. So from transcription to translation, since the proteins start forming on the ribosome, they start folding into their native structure. So initially they are unfolded state, and with the help of chaperone and the different enzyme in the body, they are take their normal 3D conformation. This 3D conformation is required for normal cellular function. So if any problem is there in this process, these unfolded protein can undergo take different conformation and can form the misfolded state. This misfolded state can also be converted into the properly folded state if chaperones and enzymes are available. If not, then these misfolded protein, they form aggregate. They have a tendency to interact with other molecule and form aggregate. Why they form aggregate? If you see this energy landscape of protein folding, you'll find that this native state of the protein, which is three-dimensional structure, they have a least energy in the native form. However, all other partially folded form, they have higher energy than the native state. But what happens if this partially folded protein, it expose or some portion of the protein which is taking the beta sheet structure. So this beta sheet structure causes more intermolecular interaction. You can just understand that for proper folding, intramolecular interaction is needed in the protein, that the interaction between the same protein peptide. However, if that intramolecular interaction is not proper, then some misfolding occurs, and it has been identified that the beta sheet structure that is formed during this misfolded structure, they have high tendency to interact with the other molecule which is having the similar structure, or they have a tendency to govern other molecule to take such kind of misfolded structure and aggregate very fast. And this final aggregate product is called fiber. We call it amyloid fiber. And if you can see that this amyloid fiber has a more stable conformation. So energy of this amyloid fiber is least. So once this amyloid fiber is formed, it is very difficult to convert them into any other form. However, oligomers, amorphous aggregate, and partially folded state of the protein can be converted into the native state. And you should remember this, because this is very important when we are talking about to use this protein folding phenomena to develop a long-acting drug. So in the when we talk about the disease and how this protein aggregate, they call disease. So when this normal protein, we know that they have a particular very specific function, if they and that is associated with their conformation. If that conformation is not there, so they either get degraded by the body. If not degraded by the body, they will either gain a toxic activity, or there will be a loss of function. Why loss of function? Because protein is not performing their function, because they are not in a proper conformation. So the function associated with that protein will be not observed in the body. Gain of toxicity means they are now cause the disease. So initially I'll take an example of how gain of toxicity is causing different diseases. So I'll take neurodegeneration is very good example, and why neurons are, you can say, very labile for such kind of aggregate, or they are very, these aggregates are very toxic to a neuron, because you should understand that neurons are the only cells in our body which are post-mitotic. post-mitotic means they cannot undergo division. So what happens? There are many proteins has been identified which lose their conformation with age and they get misfolded. This misfolded protein, if they are not degraded by the cell degradatory system like autophagy and proteasomal pathway, then they keep on aggregating and form fibers. Once this fiber and this oligomers are formed, these oligomers are very toxic, and they pose a different kind of through different kind of mechanism. They induce toxicity to the cell, and ultimately the neurons are dying. So what happens then? Because neurons under normal cells, you can say, if these oligomers and fibers are forming, so what happens? Cells get rid of it by division, because once they are dividing, the load of intracellular aggregate will be shared by the sister cell, or they can undergo apoptosis, because other cells can take up, like they can regenerate the whole organ. But in case of neuron, because they cannot undergo division and they cannot get rid of these kind of aggregates, so these aggregates pose a very toxic effect on them and lead to their loss. And once the neurons are lost, particular like function that is associated with that neuron is lost, and you can see that disease. So one of the most predominant diseases, Alzheimer disease. You all know that, and what happens in Alzheimer disease? There are like, if you see the pathology, you'll find there's a lot of senile amyloid plaque which get deposited outside the neuron, and there is a tau tangles, we call the neurofibrillary tangle, that is formed inside the neurons, and the whole neuron morphology get changed, and it affects the connection, because for a neuron to communicate, they're like to each other, they are very well connected. If they are not connected, they will lose their you can say morphology, and ultimately they will undergo apoptosis. So you can see that communication and connection is very important for neurons. If they are disconnected with their surrounding neuron, they undergo apoptosis. So what is this amyloid plaque and how they are formed? So in the brain, mainly in the cholinergic neurons, we have a protein called amyloid precursor protein, which is a you can say it's a transmembrane protein, and there are three proteases: alpha secretase, beta secretase, and gamma secretase. So if under normal condition, what happens in young age and when our neurons are healthy, this alpha secretase cut this APP protein first, and then it is cut by the gamma secretase, and then the whatever peptide P3 form, it has special function in neurotransmission. But what happened with age? Instead of alpha secretase, beta secretase cut the APP this protein first, and then it is cut by the gamma secretase. And then after this reaction, a peptide is formed called A beta 42. This A beta 42 has a very high tendency to aggregate, because it has no physiological function till now. It is assigned to this peptide, and that is why it is and having a very high tendency to aggregate under normal condition. So they aggregate and form such kind of amyloid plaque. So why this protein aggregate with aging? So if we see the process under normal condition, base and APP, they don't come into contact with each other in healthy neuron until, you can say, young age. What happened with aging? Our process of intracellular trafficking get disrupted, or what we called as a traffic jam in the brain, because traffic is not moving with the age. That occurs because of various kind of abnormalities or insult that is occurring in our neuron because of aging or some other drug or condition. So what happens under this condition? This base and APP get lots of time to interact, and then this APP cleavage occur and A beta production increases. And that is the most crucial part of the, you can say, life cycle in the neuron, when this A beta aggregate formation start occurring. If cells are not recognizing and getting rid of this aggregate, they get accumulated, and transfer, this initial aggregates are very toxic, so they are causing not only the toxicity toxic effect on the like by autocrine function, but by a paracrine function to other neuron as well. So the process of you can say if we see the Alzheimer disease, so initially we say mild Alzheimer disease when a person is having a memory loss, a lingual problem, and mood deviation. So this is the first sign of Alzheimer disease. But I would like to emphasize that disease does not start when you're seeing the symptoms. That is the problem with all this chronic diseases. Disease starts at least 20 years before when we see the first symptoms. So why we it get unrecognized? Because we don't have any diagnostic marker which can diagnose when the disease is started. Even A beta 42 formation is also not a very good marker to identify whether disease progression is occurring or not, unless we get a first symptoms of memory loss. So the disease worsen as the because when we are getting first symptom, it means the neuron degeneration has already started. So now it cannot be reversed. So we have to devise a mechanism or a therapeutic which can prevent this initial symptoms, or if initial symptoms has occurred, it can stop its progression to moderate and the severe condition where like person become bedridden and they cannot perform their day-to-day life. So various mutations has been associated with this APP protein present, like gamma secretase and APOE, but only 5% patient are familial patient. You can say mutation is associated with only the 5% of the AD patient. 95% AD patients are sporadic, means they don't have any mutation. So another disease is called Parkinson, and it is very well characterized by the word trap. Trap means tremor. You might have seen many old people who have a one side of their body shaking continuously. Rigidity, like they cannot move their different joint very easily. Echinacea, like voluntary movement is not there, and their posture and balance is changed. Like because posture is changed, so they are not able to make balance. Why it has happened? Because a part of our brain, midbrain, which has a dopaminergic neuron, in those dopaminergic neuron, what happens, there is a protein called alpha-synuclein. This protein plays a very important role in the synaptic transmission. So this protein get aggregated with the age due to some kind of misfolding there, and these misfolded protein are not getting degraded by the cells. They are not getting recognized by the cell, so they get accumulate and form Levy body, and these are very toxic and they cause death of the dopaminergic neuron, which is controlling our motor function. And that is why we are seeing all these symptoms. So I'll say that all these diseases are recognized. You can say Alzheimer was recognized now more than 100 years back. And we know so much, but still there's no drug which can treat these diseases. So till now, so many protein has been identified which are associated with the different diseases. So this is like just to show you how this protein aggregate, they are associated with different diseases. So now coming to know how we can use this protein as a tool. Because for disease, we know that they are causing disease, and now we have to identify the mechanism by which we can prevent them. So before going again, I would simplify that a protein is said wild type when it is having its normal native structure and forming, doing activity by forming complexes with the other partner. Because protein alone cannot function, it needs it work together with the team. So what happens if protein is getting misfolded? Suppose this pink one. So sometimes they get new function, or they can toxic function. So that is called gain on function. So we have seen that toxic function because of protein aggregation, which is linked with the disease. But sometimes happen that protein this misfolding, or you can say different conformation, has hyperactivity in case of some signaling event we have seen. So protein can also gain a function also. What happen if protein is lost or there's no protein? So function, there will be loss of function. So why loss of function occurs? Suppose this is a wild type protein, and if point mutation is occurring in the more structural part, so protein loses its function. Okay, because it is more making the protein more structure, because for function it need a kind of flexible domain which can change conformation with the interaction with the other protein as well. So it has been observed that when in the disordered region of the protein, there's some mutation which can so that it can increase the activity of the protein. And this is a very good example. Now you can correlate with the SARS-CoV-2 infection that is going on, coronavirus infection. There also you can see that how fast mutation in the spike protein and other protein envelope protein is affecting the infectivity of this virus. And it's a live example of to see that how these mutations, if they can change the function of that protein. So now how to treat that loss of function? So either the gene therapy in case of human diseases, or human body, if you talk about, if some protein is losing its function, what we can do, we can change the defective gene, but gene therapy is always a very tedious process and not very cost effective. Another is the protein therapeutics. So what is the protein therapeutics? Protein therapeutics is like therapeutic approach of using protein or peptide for treating some diseases. And it is one of the, you can say, most viable approach to treat any disease. And it is having many advantages over gene therapy, because each protein has a very specific activity. And they can, we can identify any protein which is linked with some disease. Now with the our recombinant DNA technology and protein expression is technology has developed so much that we can immediately express artificially those protein and use them as a therapeutics. So ease of use and like you can say other advantages of protein therapeutics, that like we can identify and use loss of function protein and use them as a therapeutic. So it can replace the, you can say, synthetic drugs. Why synthetic drugs? Synthetic drug even cheap, but they have many side effects. That is called off-target effect. But proteins, they are always having a specific function. So when we are delivering, so they will do their specific function. So it will be highly specific protein therapeutics, and it will show less interference with the normal biological process because their function is very well defined. Faster clinical development and FDA approval time. Why? Because if we are using human homolog of that protein or human protein, then it's a body's own, you can say, molecule. So it has very less toxicity and there are less regulation for FDA for approval. And if protein is in a proper functional form and given in a best way, so they do not elicit any kind of immune response in the body because it's their own molecule. So seeing the advantages of protein therapeutics, till now FDA has approved more than 130 protein and peptide for the treatment of different diseases or illness. But there are various challenges that are associated with this protein therapeutics, and that is limiting its usage for other diseases, or you can say cost. What the major problem is? Solubility, the route of administration, distribution, stability. The immune response, in case of suppose human protein, we cannot use it, so unstable. If you are using some other protein from the other organism, so it may elicit immune response. So that is the problem. Or the protein that we are injecting should be physiologically active, or so some of the protein therapeutics, they need to be post-translationally modified, like they need glycation on that, or phosphorylation or acetylation or methylation. So that is a challenge to get this post-translational modified protein and peptide to artificially generate them. So all this problem leads to the increase in the cost of the protein therapeutics. So just before going, I'll say that there are 4 different groups of protein therapeutics. Group 1, that where I work, is like therapeutic enzyme, which are involved in the enzymatic activity or regulatory activity. It is used for replacing the protein that is deficient or abnormal in the body, or augmenting the existing pathway, or providing a novel function or activity. However, group 2 molecules are used mainly for as a carrier, and or they are the molecule which inhibit protein-protein interaction, or some kind of you can say targeting effect, or by inhibitors as an inhibitor. Vaccine is a very well example of protein therapeutics, where we use you can say one epitope of whole. You can understand that the subunity vaccine is mainly the protein-based vaccine, which we use for different infection. And group 4 protein therapeutics, or you can say they are part of diagnostic aging, because now the rapid antigen test, all this kind of test, they use antigen from different organism, you can say pathogen, or when we are want to do some kind of ELISA and all, so checking the different level of hormones and enzyme in the body also, it depend on the such kind of kits. So that is the biggest industry. So before going into the how protein therapeutics we can take care of those challenges to develop a new technology, I'll just say difference between the multiple injection and the single injection. When we talk about the single injection, means we are giving the soluble protein therapeutics, one single bolus injection, and what will happen in the single these are mainly the short acting protein, like they have a very short half-life of, you can say, maximum 4 hours. So that is why we need to inject them more than once in a day, so that their therapeutic level is maintained in the body. When we talk about the therapeutic window, you can see that there are 2 dotted line. So these are the y-axis show the concentration of protein that is needed to show its therapeutic effect. So if the concentration of protein is maintained between these 2 lines, then only it will show therapeutic effect. If it is going above, that happens in the bolus injection or single injection, that sometimes the peak may go above this line, and this excess amount of protein can be cytotoxic. So whatever toxicity that we are seeing with the protein therapeutic is because of this bolus dose, like we are giving a single dose and higher dose, so it is going above, and this extra protein is toxic to the cells. So after seeing so many drawback with such kind of single you can say multiple injection protein therapeutics, people started thinking of, can we make a protein therapeutic in a way that after single injection it can keep on releasing the active drug molecule for longer time in a very controlled manner. So suppose if we are injecting by any devising any kind of method by which we are making this long acting protein therapeutics, so what will happen after single injection, it will be able to form a kind of depot in the body, and from there, that depot, soon the molecule of the protein will get released in a controlled manner in a therapeutic window range. The concentration need to be maintained in this therapeutic window. If the protein is continuously releasing in the therapeutic window, is efficacy will be very high. Dependency of the on the multiple injection will go, and it will be better patient compliance. When we're talking about the patient compliance, like a person who is going to take this protein therapeutic, they should be happy to take. Like if they need to inject every 2 hours, it is very invasive process, like they will hesitate, and it's a very painful procedure. So that is why, keeping that patient compliance, people started thinking of such control releasing formulation. And in the therapeutic window also, we need to keep the protein in their proper conformation, so that they are not showing any off target effect, that they are not binding to any other receptor other than where they are supposed to bind, and also they remain soluble. Okay, so for such devices it's very important that they should be protected from the degradation machinery of our body. You know that we have different kind of proteases, very strong proteases. So whatever device that you can say formulation you are going to develop, it should be protected from those proteases, so that depot is staying for the longer time and doing its function of releasing the protein therapeutics. So different type of product delivery method now then developed. People started you can say entrapping the protein molecule or therapeutics in the liposomes in the form of dendrimers, or hydrogels, nanoparticle. So all this, in this case, the protein is present, therapeutic is present in the product, that they are attached with the something. And in contrast to normal drug, which after intake is immediately available for activity, in case of product, they need some kind of transformation, or you can say release, and then the pro part is removed and the now drug is available to do its activity. So now coming back to the protein aggregation. So I would like to tell you that same protein, it may have only one native conformation, as you can see this rounded ball, but it can adopt in different environment, it can adopt a different misfolded conformation. And each misfolded conformation, they can form different kind of aggregates. Like this one, it can undergo an amorphous aggregate, some can form globular aggregates, some can form annular oligomer, which is the most toxic form of oligomers till now known, because this annular structure, they form pore on the cell membrane and cause the immediate cell death. Another is like highly compact protofibril type of structure, and then the amyloid fibril, which is very, very tight, and it is very impossible to release molecule from there. But under certain circumstances, we can modify this pathway of amorphous, globular, and this protofibril kind of aggregation pathway in such a way that we, from these molecule, we can get the release of the protein in the more native form. Okay, so when I joined NII, so I asked, can we, just as I said, can we do this kind of misaggregation, and can we utilize that kind of protein aggregation pathway to devise a long-acting therapeutics? So I studied many proteins kinetics, aggregation kinetics, and as I said, a soluble protein undergo oligomerization and then it undergo insoluble fiber, which is amyloid fiber, which is responsible for various protein misfolding diseases. So if we see the oligomeric pathway, so oligomerization and fibrillization. So oligomerization, I found that we can divide into two different pathway, on pathway and off pathway. When we say on pathway, means these oligomers, whatever conformation they are taking, we have no control over them, and in that particular condition is very important, because in that particular condition they will go on like towards the toxic oligomer formation and the fibrillization. However, under certain circumstances, or you can say conditions, in vitro condition of, like you can say buffer, pH, or you can say presence of some salt or temperature, all this, when you modify, you can modify the oligomerization process toward the off pathway. Off pathway means they will not form any toxic oligomers, they will not undergo fibril formation. So this off pathway is kind of oligomers which retains their structure in the near native form conformation. So then I thought of, okay, I have selected some of this oligomers and seen whether they are able to release the drug from or protein from those oligomerized, and I found, oh, they can, once they are separated from the reaction and suspended through and separated through a membrane, they can slowly release the drug molecule. And I call them supramolecular assembly of protein. Okay, so now I know that I can reverse the process of oligomerization and get the protein into the normal conformation. And why these oligomers are very good, I will let you know later. So first disease that I have selected to prove my concept, or you can say prove my hypothesis, was diabetes, which is very well known. You all know what is diabetes. It's a condition when the blood glucose level goes very high, and it is due to high blood glucose level, you see different kind of symptoms like hungry, feeling very thirsty, tingling in the limbs, blurred vision, or frequent urination. So why we get blood glucose level? What is this high blood glucose level? So for normal healthy person, the normal blood glucose level, there's two type of blood glucose level: fasting plasma glucose level and the oral, or you can say fed state. When after meal, taking meal after 2 hours, you are taking your blood glucose level. So for healthy person, fasting blood glucose level, because fasting, why I say fasting is much more important parameter to find out whether the person is diabetic, pre-diabetic, or healthy. If your fasting blood glucose level is 99 or low, you are healthy. But when it is between the 100 to 126, you are pre-diabetic. It's a condition which can lead to the diabetic condition. And if your blood glucose level is above 126, you are considered as a diabetic. So why this blood glucose level goes high? So in the healthy person, a hormone called insulin is released by the pancreatic organ called pancreas. In the pancreas, we have a islet of beta cells called islet of Langerhans. They secrete a hormone called insulin. They produce and secrete a hormone called insulin. Insulin is needed for some of the cells of our body, like muscle, adipose tissue, liver tissue, brain, and bone tissue, bone forming cells. So these cells, they cannot take up the glucose unless insulin is signaling them to take up. So this is called insulin stimulated glucose uptake. And this happens whenever we take food and our blood glucose level goes high. So our beta cells in the pancreas, they detect that and start secreting insulin. Once insulin is binding to its receptor, it's activated intracellular downstream signaling cascade, and there is a transporter called Glut4 Transpositor. These are the glucose transporter, very efficient glucose transporter. So these transporter, they remain in the cell, in the vesicle, small vesicle. So once the insulin is binding to the insulin receptor and activating downstream pathway, AKT is the main effector molecule. If AKT get activated by phosphorylation by PI3K kinase, so this direct this small vesicle to dock on the plasma membrane, and then this Glut4 translocator, they take up most of the glucose from the blood, and the blood glucose level goes down. So based on this insulin mediated glucose uptake, there are two types of diabetes. Type 1 is the condition where like it's a autoimmune disease, and mainly occur in the, you can say early onset, because it's a autoimmune disease, and mainly occur in the children less than 20 years. And it is also called insulin dependent. Why insulin dependent? Because here what happens, our own body immune cells, they attack on the beta cells and they kill those cells and not allowing them to produce insulin. So there is no insulin in the body. However, receptor and everything is fine. So once we are giving insulin, they will bind and take up the glucose. So because insulin is not there, that is why it is called insulin dependent diabetes. Another type of diabetes, which is called as type 2 diabetes, which is mainly associated with the lifestyle that we are taking. It's age dependent and it occurs with the aging. Why we are saying with the aging? Because with aging, different kind of insult that we are giving our body, a type of lifestyle that we are taking, what it does, it affects the insulin receptor protein. So insulin receptor, either the insulin receptor or the downstream signaling pathways are disrupted with the aging. And even though insulin is present there, it is binding to the receptor, but is not activating this downstream signaling pathway. If signaling pathway is not activated, then this vesicle which contains the Glut4 translocator, they will not dock onto the membrane, and they will not take up the glucose. And that is why the blood glucose level will remain higher. So can we prevent any of these two? If we talk about type 1 diabetes, which constitute around 10% of the total diabetes cases all over the world, and I would say that India is the capital of diabetes. Like maximum type 2 diabetic patients are in India, and it's day by day increasing. So if we say, so type 1, we cannot prevent. There is no way that we can prevent type 1 diabetes. However, in type 2 diabetes, which constitute around 90% of the patient, they can be prevented. How? By maintaining their healthy lifestyle, like eating the healthy balanced diet, keeping their weight on check, doing like physical activity, being physical activity. So as I said, type 1 we cannot prevent. So what is a therapy for type 1? Because it's occurring mostly into the children, and it's very difficult to see them injecting insulin all the time, because it is insulin dependent diabetes. There's no insulin, so insulin is the only treatment for such kind of patient. So insulin either available in the injection form, or insulin pumps are available, or another option that now scientists are seeing, that can we transplant whole pancreas or pancreatic beta cell, which are responsible for insulin production. So these translational, you can say, research is still going on, and people have seen that hope in transplanting these beta cells into the eye retina, or you can say in the liver, and they have seen that these cells, they produce insulin in based on the their blood glucose level. They can sense blood glucose level and produce insulin accordingly and secrete into the blood. Okay, so why I have chosen insulin? Because insulin is a very interesting molecule, and it is having a very big history. The diabetes is known from centuries like 1550, but till now, still so much work is needed to make the insulin as a frontline, you can say, therapy. And also, very important thing that it is completed its 100 years of discovery in 2021. And because insulin was discovered in 1921 by three, or you can say four scientists, Benting and Best, who received the Nobel Prize 1923 for discovery of insulin. But they had their two more colleagues, Collip and the MacLeod. They shared their Nobel Prize with them because they helped them in discovering the insulin. And they are the first who purified insulin and given its a name to the insulin. After that, insulin is the only molecule for which three more Nobel Prize has been awarded. Sanger's received the Nobel Prize in 1958 for solving the insulin structure. Palade in 1974 for how the hormone is secreted from the beta cells, and Yellow in 1977 to develop the radioimmunoassay for the insulin. And it is the discovery by these scientists, all these scientists, that has revolutionized the, you can say, insulin to develop as a protein therapeutics. And right now, insulin is, you can say, saving the life of millions of people who are having type 1 or type 2 diabetes. So how insulin is synthesized, you all know, because just for your information, I'm just showing this slide. In the beta cells, insulin is synthesized in the pre-pro-insulin form. So pre-pro, when we say, there is a signal sequence. So before targeting to the ER, the signal sequence is for the targeting to the ER. The signal sequence is removed and the translation occur into the ER. ER has a proper site for folding of insulin. Why proper folding? Because ER has an enzyme called protein disulfide isomerase, which helps in binding 3 different disulfide bond, which is needed for keeping the native insulin in the conformation. You can see that after disulfide bond formation, the proinsulin, which is present in the ER, is transported to the Golgi. So proinsulin to insulin change, the C-pep chain in the proinsulin is getting proteolytically cleaved. This cleavage can occur in the ER or in the Golgi. So after it is the final form of insulin is formed, you can say that it has a 2 chain, alpha chain A and chain B attached with the disulfide linkage. So these insulin, they are stored into the small vesicle into the beta cells. And beta cells, they also have a Glut2 receptor translocator for the glucose. And these Glut2 glucose transporter, they are very high affinity for glucose. So as soon as we take glucose rich diet or blood glucose level goes up, they sense the blood glucose level and allow the beta cells to trigger a reaction which can release this insulin granule on the membrane and releases the insulin to the bloodstream. So if we say how this insulin is stored into this vesicle, so later on people identify that insulin is stored in the form of hexamer. And this hexameric formation of insulin is very stable and it requires zinc for formation. This hexamer then convert into dimer and then monomer. And then the insulin is binding to the receptor and doing all the biological function. But what happens if any condition, if zinc is not available and this hexameric form is not formed, or insulin monomers are getting misfolded, and then this misfolded insulin molecule, they have a very high tendency to aggregate, and they can form different kind of protein aggregates. And this formation of protein aggregate in the beta cell are linked with the one of the cause of type 2 diabetes mellitus, where insulin production is little bit going less. So we have learned all this process of how all this folding occurs in the presence of zinc and all. And then we looked into the type of insulin till now developed and present in the market for therapeutic purpose. So green one is showing very rapid acting insulin, which is the normal body insulin, which has a very short half-life, and it is like when injected, it is giving a very high peak. So it may give a toxic effect. So later on, because whatever toxic effect we were seeing with the insulin was because of it is high peak. So it is going beyond the therapeutic window and giving the toxicity. So people started making the short acting, intermediate acting, and then the long acting insulin was developed. It was developed by a combination of like different point mutation in the insulin gene and mixing with the different additives. So long acting insulin is the only insulin which if you are injecting, taking once in a day, so it will not form any peak, but throughout the day, it will maintain us insulin concentration in near, you can say, therapeutic window, basal line, and keeping your blood glucose level into the check. So when I started my work, so we asked the question, can we develop a long-acting insulin which can be effective for a month, and that also after a single injection? Seeing all the properties and all the oligomers property that is formed by the insulin. So then we started our work to make supramolecular insulin assembly, we call them. So what we did, we have taken the insulin and allowed them to aggregate in different environment, and then check whether those the type of oligomer that are different time point they are formed, how their structure is getting affected, and whether if they are isolated, they can release the active monomer or not. So after lots of screening, we found out three different insulin assembly, 1, 2, and 3. So I'll later on describe what. So we found out that supramolecular insulin assembly that we have formed, 2, it is giving a very appropriate release time, and it can release the active monomer from if separated through a membrane. And this active insulin monomer, they are, why we say active, because they are biologically active. We checked on the cell line and we see that okay, they are able to induce the glucose uptake in the insulin responsive cells. So another important thing, when you are making such kind of long acting drug using the protein aggregation as a tool, so it might be possible that slightly different conformation can lead to the gain of activity, and it may show off target effect. Off target effect means there are different receptor in the cells which can bind to the insulin at different affinity, like IGF-1, insulin like growth factor 1 receptor, and insulin receptor and insulin like growth factor receptor both has a different function in the body. IGF-1, if it is induced, it causes more division of the cells. You can say it's mitotic and promote the growth of the cell and division. So we checked with the receptor binding assay, and we found that the insulin and the release insulin from our SIA2 is not having any affinity for IGF-1 receptor. However, it is binding to the insulin receptor only. And its confirmation, you can say atomic force microscope that we use, that is a very important tool, I can say, for designing such kind of therapeutic, because it gives you a kind of what kind of morphology is formed. Even at higher resolution, you can see how the molecules are organized into the that particular structure. So you can see that the insulin monomer, they are very small, teeny particle, but SIA2, you can see that there's small globular aggregate. But if you see the mature fibers, they are completely different and more dehydrated, you can say. And these SIA2 are more high, you can say, containing more, they are more hydrated and more in the, you can say, native conformation. Sorry, my slide is not moving now.
Yeah, we can see. Yeah, now it is.
So very important for this therapeutic is to see whether the kind of oligomer that you are choosing and you're thinking that it can act as a long therapeutic, whether they are able to release, if they are able to release the molecule, how long. So very simple tool that we have used. We have used a dialysis tubing, dialysis process. What we have chosen, like insulin, in case of insulin, it has a molecular weight of 3 kg, you can say. So we have taken the membrane, which has a pore size of 3 kDa. It has it can allow only insulin monomer to come out, not other oligomers. So before start of dialysis, you will find that there's nothing. But after sometimes we keep on taking the aliquot from this dialysate and check for the like OD at 280, because you know that protein, they absorb at 280 nanometer, and we just see whether we are getting some kind of pattern at 280 nanometer or not. So this graph is showing in 24 hours how the protein are, you can say in this case, insulin is released from the SIA. You can find out, we can see immediately an SIA2 is immediately started releasing the insulin. And how long they are able to release the insulin. So initially we observe only for 14 days, and you can see that 14-15 days, they are continuously keep on releasing the insulin. However, SIA1 was very fast releasing, and very fast it get exhausted. So that is why seeing the different property, and we check with the different analytical tool, what is the like molecular weight of this monomer that are released with the MALDI. And here you can see the FTIR is a very good tool to see whether the protein that is released from here is in the native conformation or not. So you can see that in SIA2, it is more near to the native conformation. So we were very happy that our oligomers are working fine and they are able to release the active monomer. So now it was another question that to long-acting therapeutic, that it should form a depot at the site of injection. So what we did, we have made intradermal, subcutaneous, intramuscular, all three type of injection into the rat and mice, and check how long that depot is existing. So we can see that we have observed that uh, whatever SIA we are injecting, wherever site, whether intradermal, subcutaneous, intramuscularly, they are forming a depot, and they are not exciting any kind of inflammatory response there. And it is a beauty of like making your whole you can say aggregate and all the process in a sterile environment. So if you are maintaining your sterility at all the places, it's the only insulin oligomer that you are, and no other microorganism is microbial growth is there. So you will find no, you can say, toxic effect of these oligomers. And they nicely form a depot at the site of injection. And that depot stays for longer time. So after that, our in vitro data shows the various key property that our SIA contains, that it's a compact condensate of the pure insulin, keep insulin in the native conformation. It is a proteinase K resistant. Proteinase K resistance is a very important, or you can say trypsin resistance is very important, because when you are injecting proteins therapeutic into the body and they are making depot, so throughout in our body, in our you can say extracellular matrix, there are so many kind of robust proteases are there. If those proteases, because it is a protein therapeutics, they can cleave our protein therapeutics and dissolve from there, and our therapeutics will not be effective. So we checked. So we found that uh, uh, with the SIA formation, we are masking the proteinase K or other proteases like activity sites. So they are not cleaving our SIA. And it, as I said, it's forming a depot at the site of injection and releasing insulin for a longer time after a single injection, because it's forming a depot. And the whatever insulin is released is biologically active, and it has no toxic effect when checked on the cell line, and off target effect. We check the receptor binding acid. So now it was time to prove in the actual animal model whether it is showing any efficacy or not. So what we did, we developed a type 1 diabetes model. So type 1 diabetes model, for student I'll say, there's a chemical which is called streptozotocin. This chemical, if we give to the like 12-hour or 16-hour fasted rat or mice, immediately one single dose intraperitoneally. So what is happening? And after that, immediately we give them the food. So what happens? This medicine is taken by the beta cells, and this medicine has a very toxic effect on the beta cells, and it kills the beta cells of the mice and rat. So beta cells of the rat, once killed, they are not able to produce insulin, and slowly after 1 week, their blood glucose level goes up. So we select the animal who are showing more than blood glucose level more than 250 milligram per deciliter. So they are in the diabetic condition. So what initially we did, we have taken different doses of our SIA, injected subcutaneously or intramuscularly, and checked which one is showing. And as you can understand that we were thinking of developing a therapy for 1 month, but we were surprised to see that our different doses were showing a different time of efficacy, because the type of depot they are forming. So here you can see that 200 microgram of SIA was sufficient to give the efficacy for more than 140 days. Okay, so other doses were not sufficient to maintain the blood glucose level to the normal, near normal. So then we conducted the detailed study with this dosage, and monitored the random and the fasting blood glucose level of these mice. You can find out that fasting blood glucose level is very well maintained in all these mice. You can see the control, it is near the control, or you can say completely normal. However, in case of diabetic mice, it remains on the higher side. Random fed, like after meal, also the blood glucose level were near the control. So we were surprised to see, we keep on monitoring, and we were surprised to see that our formulation can maintain the blood glucose level till 160 days after single injection. Okay. However, single injection of insulin, recombinant human insulin, is not able to maintain that. So animal, after single injection of soluble insulin, they are not maintaining their blood glucose level very properly. So this shows that the SIA that we have generated, it can be used for the sustained treatment of type 1 diabetes mellitus. So we check whether the decrease in blood glucose level is seen because of the insulin release from SIA, or there's some other reason. So we monitored, because in the rat, we injected the recombinant human insulin, which we use for making SIA, or bovine insulin. So we can check, because these are not endogenous insulin. So we check for the endogenous insulin for the rat, which was no, because they are type 1 diabetic, beta cells were killed. So whatever insulin present was human recombinant insulin. We use specific ELISA for recombinant human, and we observe. And we have seen that recombinant human insulin, SIA made with the recombinant human insulin, is releasing after, like when injected 200 microgram, is releasing significant amount of insulin till 140 days to maintain the blood glucose level. After that, it is going down. And you can see that these are the subcutaneous skin sections, which shows, because this oligomers, they have a very many unique properties. So one property is that they show birefringes if a dye called Congorate is used. So they show orange to yellow to orange birefringes under the polarized light. And you can see the same section which is stained with the Congorate and HNE. And you can see, when we are seeing in the polarized light, you can see that this depot is there, which is getting increased with the different advancing weeks. And no kind of, like you can see, inflammatory response or other tissue lesion we can see. So another thing was, because diabetes is related with a different kind of secondary complication, like stroke, heart damage, fatty liver disease, bone disease, diabetic foot, peripheral neuropathy. So we check whether these secondary complications is also removed, or it is prevented. So definitely they are prevented. And one of the example that I am showing is a very visible, that in the type 1 diabetic rat, you will find that they develop cataract if remain untreated. So you can see significant white crystallization of the eye crystal. However, in the control rat, you will not find any kind of cataract formation. When we are giving single dose daily to the rat, type 1 diabetic rat, you'll find that slightly the cataract formation is there, but less. However, double injection further reduce. But in case of SIA treatment, because it's a maintaining blood glucose level for longer time, both fasted and random, so there is no sign of cataract formation we have seen. So we have seen that SIA that is developed by us was ultra long acting insulin formulation, or you can say form, that can release slowly from the site of injection the native monomer, which is biologically active. So this was this work that we have published in the PNS in 2010, and this is a technology also we have transferred to one of the US company for further development in 2010 itself. So now this was the example of insulin. We went ahead to another disease. because we are talking about, we now know how to tailor such kind of aggregation process and make the protein therapeutics long acting. So we wanted to check whether we can apply the same concept on other protein as well. So we have chosen another chronic disease called osteoporosis. Osteoporosis, as the name suggests, that your bone become more porous and more fragile, and you are like more prone to fractures. So there are two type of osteoporosis: primary and secondary. Primary osteoporosis is also type 1, which is like associated with the post-menopause and mostly occurring in the women after years, the age of 50. And most of the female after age of 50, they are suffering with the post-menopausal osteoporosis. And currently there is no valid treatment available for it. So secondly is the age also associated, which is occurring in both male and female. And secondary is very, you can say, acute type of osteoporosis, which occurs because of inflammatory disorder or some kind of medication. So why, if you see the bone mass and the fracture threshold of bone, you'll find that with at the age of, when you are at the, you can say, young age, 30 to 40, your bone mass is maintained so that it is not crossing the fracture threshold. But after menopause, there is a significant decrease in the bone mass due to the loss of the estrogen hormone in the female. And soon after 65, their bone mass crosses the threshold, fracture threshold limit. And then they are very fragile, like very prone to any small fall can lead to their fracture. So how the bone formation in the bone is maintained? So in the bone, our body is maintained by the action of two different cells called osteoblasts, which is a bone forming cells, and the osteoclasts, which are the bone resorbing cells. So if more bone resorption is occurring, so it leads to the osteoporosis. Bone formation is less, bone resorption is more, so osteoporosis occurs. However, there are conditions where what happens, resorption is less, but bone formation is high, so it leads to a condition called osteosclerosis or petrosis, where the extra bone formation occurs. If talk about the therapy that is available, like most of the therapy, they target the osteoclast, like bone resorption. Okay, but currently very less therapy available which target the bone formation. So mostly the nutritional, or you can say exercise, is one of the way of like positive form to induce the bone formation. Out of this, calcitonin is a hormone which is responsible for calcium deposition in the bone. So what calcitonin is secreted by the thyroid gland. What happens whenever you take the calcium rich food, so blood calcium level goes up. So this is sensed by the thyroid gland and they release the calcitonin. This calcitonin then act on the osteoclast, like their receptor on the osteoclast, and inhibit their activity. Once the osteoclast activity is inhibited, the bone forming cells like osteoblasts, they get stimulated and they make more bone to resorb all the extra calcium from the serum. So whatever extra calcium is there, that is deposited, and now this serum calcium level is maintained. So even though the calcitonin is having the hypocalcemic effect, but human calcitonin has never been considered as a first line therapy. Why? Because it has a high tendency to aggregate. So people think of, because many women, they are not able to tolerate other kind of therapeutics like bisphosphonate, raloxifene, or hormone replacement therapy. So they are not like showing any improvement in their bone mass with this therapy, or they are very toxic to them. They are showing some other toxic symptoms. So calcitonin is only option left with. But how to use calcitonin? Because human calcitonin cannot be used. So that salmon calcitonin has been checked for their therapeutic activity, and they have shown that this salmon calcitonin, they are very less aggregation prone and they have longer half-life and greater potency. So salmon calcitonin has been introduced as a therapy for osteoporosis in the both injectable and the nasal spray. But still it is not considered as a frontline therapy. Why? Because being out like not the human protein, it may cause immune response, and due to the immune response development, it will not be effective after some times. And the also the side effect that they have, injectable one, it is causing nausea and GI discomfort. Very important, like initial facial flushing and dermatitis is very strong side effect. But pruritus and at the site of injection was also very obvious. In case of nasal spray, rhinitis is very obvious. So that is why it is never considered as a first line therapy. It was always considered as a second line therapy. However, in the second line therapy also, it was not giving very good result. So what we did, similar to insulin, what we did, we have studied the aggregation behavior of the calcitonin, human calcitonin. And then after lots of research, we make the chosen a oligomer which are showing all the property that in the SIA it is releasing calcium monomer. So all the experiment we have done, and we found out that named as supramolecular calcitonin assembly 1. If you see the release kinetics, you can find out that this is the only assembly that we have observed which was releasing calcitonin in a continuous or sustained manner. They had inhibited the osteoclast activity. Their structure was also hydrated, and as you can see the AFM picture. So when we injected into the post-menopausal animal model of osteoporosis, so we found that the serum marker, TRAP5B, is a serum marker for you can say bone resorption, like whether more bone resorption is occurring or not. You can see that when the animals are treated with the SCA, TRAP5B level is going down. So it is showing that bone resorption has stopped. Then we check the bone formation parameter like bone morphometry. We check whether the more number of cells, bone forming cells are not there. So more bone forming cells are there. Then we check what is happening to the bone microarchitecture, like bone quality and parameter. So we can see that in case of ovarectomy, lots of bone loss is there. But when we are treating with the different kind of calcitonin or in the normal condition, you can see that bone microarchitecture are much better. And after SCA, the architecture improved. The bone density has improved. So in case of SCA, we were able to achieve the release only after one injection here also for only 28 days, which was good. And not only for the therapeutic purpose, I also say that we can use this SCA as a tool to understand how human calcitonin works in the body. Because earlier, whatever calcitonin people were using to understand the physiological function of calcitonin were not effective, because they were using the soluble calcitonin, and it was getting aggregated when they are injecting or trying to understand some physiological function. So always whatever concentration and you can say effective concentration they were reporting was little higher. So third example was rheumatoid arthritis. For that also you know that it's an autoimmune disease. Here what we have done, we have taken protein therapeutics like IL-1 receptor antagonist, which is also called Anakinra. Here what we have done, we have this protein Anakinra was not getting oligomerized very easily. So what we did, we used a small tetrapeptide linker, which is known. We call it that tetrapeptide we have developed, which has a very high tendency to aggregate. So we tag this protein with that tetrapeptide and oligomerize this, and then we see whether this protein can release in a significant amount or not. So we can see that this multimelic IL-1RA can release significant amount of IL-1RA like antagonist when given at 150 and 200 microgram at dose. So we developed the model for rheumatoid arthritis, and we have seen injected all this along with the Anakinra to see the efficacy. And you can see that in case of multimeric IL-1RA antagonist, the efficacy, like disease of rheumatoid arthritis, is going down. However, sorry, in case of soluble protein, soluble IL-1RA, it is effective for sometimes like 42 days. After that, it is stop showing any effect. So these are the structure of multimeric IL-1RA, and you can see that in X-ray image, you can see that rheumatoid arthritis, the swelling of joint is one of the phenomena that you can note, and you can see that that swelling has gone in after treatment. So now you can see that we have used three different disease, three different protein, to show that how we can gain another function from this misfolded protein to use them as a tool to devise a long-acting protein therapeutics. And once they are characterized properly and selected properly, they can be used as a single injection for a long time. So I think with this I will stop, because and I'll be happy to take any questions if anybody wants.
Yeah, thank you, madam. Yes, now it's towards the audience. If anyone have any questions, you can unmute yourself and pose a question. Or maybe you can type in the chat box so that the questions can be taken.
Dr. Sarika, this is Meenakshi here. Thank you for the nice talk. It's a naive questions. When there is a more of a, this calcitonin, then or there is an overgrowth of bone, then what happens?
No, actually overgrowth does not happen in the postmenopausal woman. It may be possible that at some bone area growth is occurring more, but that could be a different condition. It varies from person to person. But calcitonin, the dose that actually in the human we don't know, but in the mice or rat model that when we used, so there we did not find any kind of you can say abnormality in the growth pattern. Definitely we have seen because here bone biology is a very vast and I think that this undergrad and postgraduate student, they were not be knowing much bone biology. That is why I did not expand it.
Right, yeah, that's what.
Yeah. So what happened, there are two type of bone, cortical bone and the trabecular bone. So we have seen that the calcitonin, till now it is not reported how calcitonin acts on the bone, but we found out that calcitonin is acting more on the this cortical bone, which is thick bone, making them more stronger. Trabecular bone is the like inner woven bone, which is metabolically you can say metabolically more active. But calcitonin is having specific effect on the cortical bone. That is why the picture that I have shown is the cortical bone structure, because we did not find any kind of difference in the trabecular bone.
Yeah. Fine. Thanks.
Madam, I can ask one question.
Yeah, sure.
This protein aggregates, are they naturally occurring as a therapeutic, or will have to isolate them and then they can be used as an...
Actually, it's a good question. And first hint comes from the body only, because many, as I said, insulin also stored into the cell in the hexameric form. So what I observed, that many hormones in the body, formation of such kind of aggregate is a biological phenomena to keep the protein in the native conformation and in the more you can say compact condition. Many like growth hormone, there are some other hormone. If you see, you'll find that protein, those hormones are stored in the form of such aggregate only.
Okay, yeah. But many of these folded or unfolded or misfolded proteins, they are usually degraded, no, with the help of a protein.
No, if they get degraded, then disease will not occur.
Oh, okay, yeah.
That is what disbalance we say. That is why these are age dependent, because with age dependent, as the age advances, what happens, the protein like our cellular degradatory process, they go down, and then is a like a balance between the production and degradation goes toward more production and less degradation, and then they start accumulating and showing their toxic effect.
Okay. Yeah, any questions from the audience? Dear students, if you have any question, you can please ask.
Ma'am, I have one query
Yes.
regarding your SIA insulin. So,
Yes.
like, have you analyzed its efficacy, with respect to the age-related manner? I mean, like,
Type 2
uh,
diabetes.
yeah, type 2 diabetes.
Okay. Actually, we developed for type 1. Why? Because insulin was not there. And when we develop for mainly the kids because there's no insulin. Type 2 diabetes is a very complex disease. And insulin is not only therapy for them. We do checked in the animal model of type 2 diabetes. We did see some good response, but I would not say it is as good as in type 1, because there the disease pathology is very different. Insulin is already there. They are hyperinsulinemic. So giving more insulin is we have seen definitely it is giving the beneficial effect, but it was not a very good you can say mode of treatment. It's a last option for type 2 diabetes to inject insulin.
Okay, and the oligomerization conditions like would be variable from protein to protein. Like,
Yeah, yes, that is the beauty, like a skill that you have to develop. You have to understand your protein. You have to feel your protein. Then only you will be able to. That is what I am saying. Na? Even the salt concentration, buffer concentration, pH, and temperature, whether you need to shake it or not, all this matters.
so, ma'am, like when we are developing therapeutics and like when we are developing injections from like those SIA, so like I mean, how efficiently like one can handle it?
That is what I'm saying. It's not easy. Definitely, if a person has, unless they have a passion, okay, you need to have a passion for developing such new kind of therapeutic, developing such long time acting therapeutics, because you have to keep your eyes, you have to observe each and every event. So suppose for insulin
Ma'am,
also, suppose
your
we
voice
are,
is cracking, ma'am. Can you please repeat it once more?
hello. Okay, so I just wanted to say that all the conditions like buffer condition, salt condition, temperature, whether you need to shake it or not, keep it in a stagnant form, all this matters when you are talking about developing such protein therapeutics, and it varies from protein to protein. So for particular protein, you have to optimize each and everything. You have to take out randomly different oligomers. As I said, you have to see their release kinetics. So not the dialysis membrane, because it's very difficult to handle very small quantity. You can use a small append off, cut the you can say tip, and cover it with the dialysis membrane. So it makes a even PCR tube, small PCR tube, if you have a very small amount of protein, because for screening you cannot take much and take see the release kinetics. So those kind of setup really good to initial screening of your oligomer, which can be used for further studies.
Ma'am, like one more question. Sorry, like your work is so interesting. Actually, like I mean, when we are talking about releasing kinetics, so like, can we have like differences from model to model? I mean, from animal to animal. So since you have used mouse model, but then like we are
Yeah,
developing
no. Here I have shown only the rat model I have shown, but we have studied in the rabbit as well model.
yeah.
So kinetics definitely it was in the rabbit. It was not showing for 140 days, but definitely it has gone for above 100 days. So depending on that is what the dose optimization from one model to higher you can say rodent, when you go, you have to optimize that. And even for human also, for each and every drug, we have to optimize the dose. We have to see at which concentration
Excuse me, ma'am.
injection will give you a better result. Yes.
Excuse me, ma'am.
Yes.
Ma'am, can I ask a different question related to protein?
Yes, please go ahead.
Ma'am, actually, I want to know that many people use this kinds of means a bodybuilder uses protein to means for so how much quantity should a person consume a protein means according to the body weight.
uh, uh, it's a good thing, but I'll say that over protein will always give you kind of problem, because it will be toxic. Why? If it is in the, what happens in this supplements, first of all, they are not in a very pure condition. They always have a lots of salt along with them. And secondly, they get absorbed very fast in your body. And body, if it is not getting metabolize it, so it may create kind of other complications. Okay, so each and every individual should not go for how much protein they need. They should look for how much protein they can easily digest and they are not seeing any kind of problem. So individual to individual, this supplementation is given like you should take this much. It depends on a like initial observation what they have seen in the healthy person. But every person, it cannot be applied, because you know that we are very heterogeneous population, so everybody's body is different. So for each and every nutrition, I will suggest that each and every person should observe. They can observe themselves very quietly ki what is happening, whether my body is getting benefited what I am taking or not. So definitely over protein doses will affect your liver as well as kidney. And if you're not properly hydrated, it will not get excreted out by the body. And you have to take supplementation for keeping your liver and kidney also healthy by and keeping you hydrated, so that if any ill effect by the overdosing of such supplement is occurring, so your organs are not getting affected. So the day you will stop like you are taking only supplement and not giving other supportive thing to your body to keep them all the organs active, so definitely you'll see the toxic effect.
Ma'am, can I ask a question?
Yes.
Are there any side effects of excessive amount of protein consuming?
No, that is what I'm saying. I just said that definitely for any amount of protein that you're taking, it should get metabolized. If it is not getting metabolized, then only it will cause problem or toxicity. So for properly getting metabolized, your liver and kidney should also be healthy, and you should be like hydrated, keep your body hydrated, so that these two organs, they can excrete out or metabolize your ingested protein very well, and it can be utilized in the body properly. If no more question, can we move ahead with the quiz?
Ma'am, there is one question in chat box.
Okay.
Many times we see kids have difficulty in remembering things more frequently. Is it a neurodegenerative symptom?
No. See, memory formation is a very complex process. As we say, the more we practice, the more we'll become the perfect. So long-term potentiation is a process which is involved in the memory formation. So if after lots of training, still child is not able to remember something, then definitely it is associated with kind of Down syndrome or autism. So you can check for that, but give them a proper environment first. If you're training them, make sure that the child is paying attention to that what you're trying to teach or trying to make a memory for him. If after that also child is not making any kind of memory, then only it's a problem. It could be a genetic problem or some other behavioral problem.
Any more questions? I see one question. Does pancreas transplantation work efficiently for type 1 diabetes?
I would say I have been working with the Diabetes Foundation. It's in University of Miami. So that is the only institute which are working. They are working on the transplantation of this beta cells, islet, and they are targeting liver as well as retina. Why retina? Because the retina is the only area of the body which is where the immune cells cannot attack. You can understand, if type 1 diabetes is a autoimmune disease and body already has a antibody against the insulin, and if you are transplanting somewhere, the process of again aggravating the autoimmune disease will be higher. So they are working on it to how to suppress the autoimmune response and target the organs where the immune response will be less, but insulin can be secreted based on the like blood glucose level. So they are also like based on our this protein aggregation long term SIA. So there are many companies, they are further developed, and they have make this into the kind of wearable membrane. And there they are saying ki with the glucose sensor, it can release insulin in more sustained manner, even for the longer time. They have shown that it can go on for a year. We have shown for 3 months, but they have advanced our technology to years. So, but still, I have not seen that any company who is making such kind of you can say devices.
Yeah, we'll take one last question.
I'll also add that for this pancreas transplantation, so in human, people are trying the pig pancreas beta islet cell or from the bovine, because for that they are thinking the immune response may for sometimes they can escape. But definitely the eye retina is one of the site, and the liver is one of the site that they are targeting. Some cases they are targeting intramuscular also. So soon, like many, I know that many positive cases, they many kids who are having type 1 diabetes after beta cell transplantation, they are now having a better life without any injections. So still lots of research they are doing, and I hope that another 5-10 years this transplantation will be very useful.
I will take one last question and then we'll move to the quiz.
Yeah, sure.
Is protein folding also responsible for autism cases?
Yes.
Okay.
So as I said, till now more than 30 proteins are known which can lead to the central nervous system related diseases. Because as I said, why our neurons are so vulnerable to such kind of aggregate? Because they are post-mitotic. They cannot get rid of these aggregates unless their like glial cells, or you can say their own degradatory pathway in the neuron, they are taking care of this aggregate. So that is the major reason, and that is why you see more disease in when the neurons are like matured and they are not dividing.
Okay. So we can go for the quiz. So how we are going to plan it?
So I'll show in each slide I have one question. So student, they can respond into the chat box, and Jyoti will record that, right, Jyoti?
Yeah, ma'am. Actually, the answers, correct answers would be calculated on two bases. One is the correctness, and the second is the promptness. So please be prompt while typing your answers, because I'll only take into consideration whoever is answering it correctly and very promptly. So yes, ma'am, you can start.
Okay. So now it's a quiz time. So first question is, which peptide is associated with Alzheimer disease pathology?
Ma'am, if that is there in the slide, so you can please present the slides so that they can read the questions also.
No, it's fine. Jyoti, is visible?
No, ma'am.
No.
Okay.
I think one answer is there.
My slides are not visible, Jyoti.
No, ma'am.
No, no.
Oh, okay, I'll again share my slides. Okay, just. Okay, I stopped sharing now, so I'll just open and then I'll again share. I think I lost my own connection. So. Now it's visible.
Connected with two devices, I think.
What happened?
Ma'am, you have connected with two devices, so your voice is echoing.
I'll take care of that.
Just log out from one.
Anyway, I am muting. You can just go ahead.
And there's a lot of echo.
Here it is not visible because I am in the sharing mode.
From this, it's not visible.
Okay.
Or you can do one thing. Can you type the question in the chat box?
Just a minute. Just a minute.
Do you want me to do it?
I'll try. I'm trying again. Just a minute.
Okay.
It is fine now?
Yes, ma'am.
Okay. Okay now?
Yeah, it's visible.
No, ma'am, but it is better if you can put it in the from the current slide. If you can select that mode, slideshow.
No, it's a slideshow mode only.
No, we can see the other slides also.
No. Still you can see.
Well, it doesn't matter as long as we are able to see the questions.
Yes, yes, yes. I think
yeah. Because in my case it is showing the full screen.
Don't worry, Dr. Sarika, it's okay. Like, let's read out the questions.
So first question is, which peptide is
that is already done. Let's go to the next one.
Okay.
Which protease?
No, ma'am, the answers are not there. Ma'am, for the first questions, there is no answer in the chat box.
No, I saw then beta amyloid, everybody is written.
Okay.
Yeah, it's there. Lot of people have written already, Jyoti.
Okay.
So let's move to the next question.
Reshma Nalange. Yeah, she has answered.
No, a few more have also.
Yeah, they have, but ma'am, we'll consider only the first one.
Yeah, that's
right.
Okay,
now student be active. The second question is, which protease enzyme involved in the production of A beta 42 peptide?
Ma'am, can you move the slide because it's not visible?
It's not coming, ma'am. Okay. Shivani Singh has answered, and others, please do answer.
Can you repeat the question?
Which protease is involved in processing or producing the A beta 42 peptide, which is pathogenic?
Shivani has answered first. Ma'am, again, if you can put it in the slideshow mode, yeah, that would be better because no, they are not able to see the question. I mean, though two questions were visible, but now for the next, they won't be able to see the question. Yeah, now it is fine. Yeah. So for this, Shivani Singh has answered, and the answer was correct also. Can we go on this third question, ma'am?
Okay, third question is protein that form Levy bodies in Parkinson disease. Please name that protein.
Okay. I think Sneha Yadav has answered it correctly. The full name she has answered.
Okay. So can we go to the fourth question?
Yeah, ma'am.
The fourth question is insulin dependent diabetes is called what? Type 1 or type 2?
Type 2.
Please remember insulin dependent diabetes.
Okay. For this, I think, ma'am, Komal Rani has answered it correctly.
Okay, so can we move to question 5?
Yes, ma'am.
Which cells of pancreas produce insulin?
Sanika Deshmukh. Sanika Deshmukh.
Okay, so can we
Beta
move
cells and islets of Langerhans both are correct answers.
Correct answer, yeah. So next question is, who discovered insulin and when?
Okay, Komal Rani.
Oh, Komal is very active. So can we move to 7th question?
Yes, ma'am. It is the answer is Benton and Best in 1921.
Yeah.
Yeah.
What is the full form of SIA that you have heard today?
Answer very quickly. I mean, don't go and Google it. Just answer it.
You will not get it on the Google. Sorry.
Okay. Supramolecular insulin assembly. Shalu Nair has answered.
Okay.
Please answer. Shalu Nair has answered it now.
Okay. So we are moving to question number 8.
8. Yeah.
What is the fasting blood glucose level for a healthy person? For this, you have to consider within 10 seconds if somebody is answering. Otherwise, they'll Google and answer.
But all the answers I'm seeing are okay. Manasvi Budkar has answered it. 99, 99, 99 and below. I think 99 and below is the right answer.
Yeah, answer.
Shalu Nair.
So next question
They
is
most of them have answered 120.
Okay.
Yeah.
Fasting blood glucose level for diabetic person. What is when we consider them diabetic? So what fasting blood glucose level is there?
Okay. Divya, 126 or high. Yeah, this is the right answer. 126 or above, right? Ankita has
Yes.
answered 126, but I think, ma'am, the correct answer should be 126 or high.
Yeah.
Yeah.
Because higher than 126 is considered diabetic.
Yeah.
So next question is, how many disulfide bonds are there in the active insulin molecule?
This is the last question. Please answer
Yes.
it.
I have asked very basic question, which is part of like your PSC, MSc course also, except one or two questions.
Okay. Okay. Two. They. Oh. Okay. But, ma'am, they all have answered
Two.
two.
No, answer is 3. Huh?
Okay. One. Okay. Then three.
And now he's saying,
Ketan.
yeah.
No. For this, I don't think we should give.
Ketan is the right answer. Ketan has given
No,
the right
ma'am.
answer.
But after hearing the answer, that three was typed.
Okay,
That
okay,
I.
okay.
So, yeah. So also with this, I want to request all of you, if you can switch on your camera, I'll take a screenshot.
Yeah, that is what I was also going.
Sir, also after the meeting, please send me the recording.
Yeah, I'll share it.
Yeah.
All of you put on your videos. Still, we got a very good response.
Yeah, it was good.
Yeah,
Hope you all like the lecture and the message that I wanted to convey, that all the process, biological process, they are very interesting. If we understand them very correctly, we can use them as a tool for the devising some kind of therapeutics or anything. So it is very important to don't understand that this process is not good, it is only disease oriented, but at some time point, if you can correctly understand, you can use it as a tool and make something useful.
So some more screenshots I need. I would request most of you, if you can switch on your camera here.
It was an interesting topic, somewhat different, what we could hear.
It was, I think it's a different topic that I have taken, but I hope students liked it. How to play with the protein and their structure.
Yes.
It is more like playing. The people only who are playing with the protein structure and the like understand, feel it, they can even devise even better. I can say therapeutics.
Yes.
Hello, ma'am.
Hello.
Ma'am, can we get the recording of this meeting?
Yeah, you can contact me. I'll send you the link.
Okay, ma'am. Okay, sir. Thank you, sir.
Okay. Yeah. So yeah, we have taken the screenshot, and now I think we can wind up.
Thank you, Jyoti.
Okay, thank you everyone for listening to me very patiently.
Thank you, ma'am.
And you can contact me anytime if you want to know more about my research. Okay.
And I also want to tell all the students as we are winding up that the PhD applications are on. Like we are having the PhD program with us. So you can go to the website and you can check. We have extended the date also. So it would be a good opportunity for all the students if you can go and send your applications for PhD programs. Sir, I'll send you I think I've already shared the infographic with you. And again, I'll send it to you because we have extended the dates.
Okay. Yeah.
For application, so that you can share with your students, and they can, if they feel like, they can apply for the program.
Yes, sure.
Thank you, ma'am. Thank you, sir, for nice session.
Kalpana, madam.
Yes, sir. Yes, sir. So let's aggregate all information aptly in our brain. I hope it would be a brainstorming morning, and a wave of heat would have generated into this cold weather to make more warmer. So it's my privilege to propose a vote of thanks on the behalf of Vidya Pratishthan Arts Science Commerce College. I extend a very hearty vote of thanks to honorable delegates from National Institute of Immunology, Meenakshi Madam and Jyoti Singh Madam, to make this webinar more successful. My special thanks to our resource person, Honorable Speaker Dr. Sarika Gupta Ma'am, for her wonderful presentation about research updates in the protein aggregation and like protein therapeutics, especially about SI. The knowledge she shared with us would be the chaperone for correct folding of students' understanding regarding the scope of biomedical research. I express my gratitude to our principal sir, Dr. Bharat Shinde, for providing continuous enthusiastic support to all such events. Thanks to officiating principal Dr. Ghadge sir, vice principal Dr. Kashi sir for encouragement. My deepest gratitude to Pendharkar madam for illustrious welcome speech. A very hearty thank to Dr. Bose sir for organizing such a catalyzing event to commemorate Azadi ka Amrit Mahotsav. All teaching and non-teaching staff for participation and support. Lastly, thanks to all audience for their active participation. Thank you all.
Yeah, thank you. A special thank to Jyoti madam. Many a times it happens that we have to postpone it.
Yeah.
Yeah, due to some technical.
But Jyoti is always persuasive and she makes things happen.
Yeah. It's at one point it happened so that we'll miss this chance to have a lecture. But then she gave us a second chance. So
no, sir, it's fine. It happens like, and we'll be sending. So please send me your postal address. We'll be sending certificates to the students who have answered the quiz questions correctly and promptly. So I'll send you the hard copies of the certificates. So please, after the program, send me your postal address.
Yeah, sure.
It might take some time, but we are definitely going to do it.
Okay, fine. Thank you.
Okay. Thank you, sir.
So
thank you so much
to all of you.
Thank you, ma'am.
Thanks to everyone.
Yeah.
Thank you, ma'am.
Thank you, ma'am.
Thank you, everyone.
Thank you, Bose sir.
Yeah, thank you, madam.
Thank you, Kalpana ma'am. Thank you, Meenakshi ma'am too.
Thanks to all the students.
Yeah, thank you, sir.
Yeah.